So thanks. I'm Tim Paulitz, a research plant pathologist with the USDA ARS here in Pullman, and I've been here for almost 25 years, primarily working on soil-borne pathogens, fungal, and nematodes. But about 10 years ago, with the development of new technology of being able to look at the microbiome, I started being interested in soil health and plant health and microbiome of wheat. And we did a lot of work on studying that years, primarily because microbes provide so many benefits to the plants in terms of nutrient cycling, making nutrients more available, in terms of nitrogen cycling, in terms of carbon cycling with fungi degrading things, and also the role that microbes play in suppressing plant diseases. That's been a really important part of our research in our unit over the last few years. But we don't grow wheat in monoculture in most parts of the state. And I became interested in the effects of the previous rotation crop on this microbial community. Um, and one of the reasons that I was really so interested in that is because we can think of kind of a legacy effect. So we grow a crop one year, and that crop will select and increase and maybe decrease other groups, kind of select its own microbiome. And then when we plant the wheat the next year, that wheat will be exposed to that microbiome that developed in the previous rotation crop. So that's kind of the reason why I'm interested in what effect rotation crops have on wheat. Okay, so let's see. So anyhow, and a lot of this is going to be focused on canola, because one of the things about rotation crops is we don't have a lot of them. There's been a lot of interest in canola for the last few years, and a lot of reasons for this. Canola acts as a break crop for pathogens, allows rotation for herbicides. The large taproot can break up compacted soils. We've got the biofumigant effect because of the glucosinolate background. And in general, a lot of the research has shown that canola can have a positive effect on the subsequent wheat yields. The other dynamics that's happened here in the last few years, of course, is we've had good canola prices. We now have a crushing plant in Warden that can handle all the canola. And the other thing that's happened over the last few years is there's been an increase in canola because of the Roundup Ready canola being used to control the herbicide-resistant grassy weeds, especially Italian rye. Let's see, I guess I'll have to use this. And if you just look at the production of canola over the last few years, you can see it's really taken off. You know, for many years it was around 20,000 acres, 40,000 acres, but this last few years it's really taken off quite a bit. And so what I'm going to base this all on is a story that really started 10 or 15 years ago with Bill Schillinger and Jeremy Hansen, and they were interested in the rotational effects of canola. And they set up this study with Hal Reardon in Reardon, or Hal Johnson in Reardon. This is land that had been in wheat for over 140 years with no rotation. He usually fallowed it once every 3 years. So he set up a rotation of winter canola, spring wheat fallow versus winter wheat, spring wheat fallow. And Bill was interested really in doing all the agronomics of this thing and seeing what the benefits were. But the surprising thing he found after that 6-year study was that we were getting yield hits. And this just shows a picture of one of those fields. So this is spring wheat after winter wheat. And on the right here, we see the spring wheat after winter canola. And so this is very perplexing to us. We ruled out water, we ruled out weeds, we ruled out pathogens, we ruled out a lot of other agronomic effects. And so this led us with the hypothesis then, could this be due to the microbial communities present in those rotations? And the only clue we really had was that Jeremy Hansen, as part of his PhD, did phospholipid fatty acid analysis. And this is a way of looking at broad microbial groups based on these fatty acid markers in the membrane. And the one thing that was really consistent was that both the fungi and the AMF fungi were consistently lower in that winter canola rotation. So I don't know if you can you see my arrow here? So basically this first bar here on the far left shows the winter canola, and then this is the winter wheat, and then this is the winter spring wheat following the winter canola, following the winter wheat. So you can see consistent reduction in the total fungal biomass, and the same thing happened with the AMF fungi. And I'll talk about the AMF fungi later on. These are important um, uh, symbionts on plants, but surprisingly or not surprisingly, brassicas are not mycorrhizal, unlike most of the other plants. And so, but unfortunately, we didn't have any DNA at that point. But he did have DNA on another study. That was a two-year study that was done in Ritzville, where he sampled winter canola and winter wheat, and we extracted the DNA from the rhizosphere, and then did this amplicon sequencing, basically trying to look at all the fungal and microbial communities. And the question we had was, can we detect any differences in the microbial community between the wheat and the canola that may explain this yield loss? Is there one group that was more dominant on canola than on wheat that could be detrimental to wheat? Or does the canola, on the other hand, suppress a major component of the microbiome that could promote the health of wheat? So it was kind of a fishing expedition to see if we could find this smoking gun, so to speak. But the surprising thing was when we looked at the microbiome of those two crops, we found that the crop itself had a really very minor effect on the communities. In other words, the bacteria and fungi on the root were pretty similar. The biggest effect was location, and this had a stronger effect than crop. And Ritzville, the squares are so that's where the differential happened, but we did not really see much of an effect of the crops themselves. And then another way that you can look at this is something called a heat map. And so basically what this is showing is all the different bacterial groups in a row. And then each dot or pixel is one of the samples, and the abundance in that sample is shown by the color. So the ones that are bluer are lower abundance. The hot colors, the reds, are higher abundance. And what I did was here, I put all the canola on the left side, all the wheat on the right side. And if you just look at these and stare at these, you can really see that there isn't any really difference that you can pick out between the two sides. We find a whole big group down here of groups that are abundant on both of the plants. The only ones that we found difference was up here, and I've kind of put a circle around it. There's a few groups that look like they're higher on the wheat than the canola. And these are Pseudomonas, Flavobacterium, and Pedobacterium. We know already that those are big components of the wheat rhizosphere. But that was the only difference that we saw. And the other thing you can do is you can do this differential abundance. So you can kind of separate out of all those thousands of OTUs or groups, which ones are more highly abundant in one than the other. And so this is looking at Ritzville in the fall. I put the ones that are more abundant in canola on the left side, the ones that are more abundant in the wheat on the right side. But the take-home lesson here is that these are all mostly red dots, and that corresponds to actinobacteria. So those are a type of bacteria that are spore formers, really adapted to dry conditions. And that kind of makes sense when you plant in the fall. That's what you're going to recruit. And then if you come back in the spring, we can see it's kind of shifted. We're seeing a lot more of these purple groups, which are Bacteroides, another big group there. So there seems to be this shift from fall to spring in terms of what's the sphere. So again, strong seasonal effect, mostly actinobacteria in the fall. These like to survive the hot, dry conditions. Whereas in the spring, we find these fast-growing things that are probably growing on the fastly expanding roots that are pumping out exudates, etc. So in the end, we didn't find any smoking gun with the bacteria. Fungi, similar experience. We found, again, we could separate them out by location. And then when we did this similar heat map, again, lots of them that are present on highly abundant on both. The only thing we did pick up, and I put them with these other red circles here, there were a few groups that were more predominant on the wheat, and it turned out that those are potential pathogens. So like Typhula, snow mold, Thanatophorus, that's Rhizoctonia, and then Microdochium. So that's about the only thing we found. The presence of some of these potential cereal pathogens. And that was kind of good to know. So it tells me that the method is working. So again, these are the 3 or 4 different pathogens that we detected that were present on the wheat. Okay, so again, no smoking gun at that point. take-home lesson seems to be canola and wheat share a lot of common bacteria and fungi in the rhizosphere. So then we started another experiment in 2018 at Ron Geray's farm, and this was a long-term no-till site that Bill Schilliner has been running since the late 90s. And so he had this rotations already present. So we said, okay, let's take a look and see whether we can see that similar effect here. So the first year we would have either winter wheat, spring wheat, triticale, or winter canola, following the second year with spring wheat, and then following the third year with the fallow. And so our hypothesis was that the crop rotations would affect these microbiome on the subsequent wheat crop, that maybe there's some pathogens promoted by the canola, and also this idea that based on Jeremy's work, maybe the canola is reducing the abundance of the AMF fungi. And we found, in fact, that the canola did have a big effect. So here's looking at the inside the root. We call it the endosphere, outside the root. The blue samples are all spring wheat following winter canola, very distinct. And then we find kind of a clustering of all the ones where we had the cereal crop in rotation. The other interesting thing was these red dots. This is actually a continuous spring wheat that he's planted for the last 25 years. So that was also very distinct. And that kind of makes sense since that wouldn't have been in rotation with anything. And then we can do the similar thing here, looking at what's more predominant in one than the other. We did find with the canola that we were picking up more actinomycetes on the canola side than on the wheat side. But the interesting thing, oh yeah, so then we'll look at fungi. Fungi, similar story. The canola rotation and these teal or blue separate out nicely. In this case, the triticale, which is the kind of green here, also seems to have selected for a significant community. And then when we do that differential abundance thing, we find there's a couple of potential pathogens that show up as being abundant on the canola side, Wadia cercinata, which we know before from some of our other work. But the other interesting thing was that on the abundant on the wheat side, we found glomeraceae, which are arbuscular mycorrhizal fungi. So that was another hint. Again, this AMF thing seems to keep coming up and up. And then one more piece of work that we did from the ITS sequence, we found one group. This is Rhizophagus. This is a genus of AMF fungi, and you can see that it's really suppressed in the winter canola treatment, which is this olive one here compared to the others. And so at that point, we said, let's look at mycorrhizal fungi, and these are symbiotic with most land plants except Brassicaceae. They colonize the root and they put out hyphae that extend out and help it take up phosphorus and other immobile nutrients. And I think there's also some evidence that it can provide disease and drought tolerance. And so this is just a diagram showing that. This is a cross-section of a root. They infect the cortical cells, produce these arbustles, and that's where the exchange occurs. So what's happening here is the mycorrhizal fungi are taking up phosphorus and exchanging that for carbon inside the cell. So again, these are obligate symbionts. They're completely dependent on the plant to provide them with carbon, mainly fatty acids. Here's some other microscope slides. Again, showing a cross-section of a root. These big structures here are called vesicles. That's where the fungus stores a lot of the lipids. And then this is showing a close-up of these arbustles, which are actually inside the cells, but do not penetrate the cells of the plant. They're essentially right up against it so they can exchange. Incredible structures, highly branched, and very short-lived. These can only be in the cell for maybe 2 or 3 days, and then they get recycled. So we then used AMF-specific primers, which could give us better resolution. And what we found was that there's probably about 6 different genera out there. Some of them are very highly abundant, very high frequencies here. So what I'm showing here is a frequency distribution. So these four or five are very abundant, and then you've got a lot of these minor ones that are very, very low abundant. And then the other interesting thing when we analyze this data was that what we're looking at here is the sequence counts versus the different treatments. So the higher the bars here, the more sequences that we identified. So here you can see, for example, the spring barley, the spring wheat treatment. But then when we look at the winter canola treatment, there's a lot of them that are pretty abundant in the roots, but a lot of samples didn't have anything or hardly detectable compared to the other. And that's another indication that the winter canola treatment is really suppressing the infection of that subsequent spring wheat crop. 2020, similar results, except not as clean. Here we saw some triticale and some winter wheat that also had low yields. And so presently what we're doing now is we're going back and doing specific AMF sequencing, but not only the following winter wheat crop, but we also want to look at the rotation crops themselves. And so the key takeaways then are that we've identified that rotation crops can significantly shift the microbiome of wheat. We find that actinomycetes seem to be enriched after winter canola. And I'm really intrigued by this because a lot of these actinomycetes have been shown to play a role in drought resistance. And so that's something that we're going to be looking at as part of some projects. There may be some potential unrecognized pathogens, but I think the big take home here is that we find this decline of AMF fungi following the non-host. So another take home is that I think these microbial legacies will be important in considering designing crop rotations. And then these are some of the future research questions that we want to look at. I'm still intrigued by mycorrhizal fungi in dryland wheat because the dogma has always been that you get the benefit when you're in low phosphorus conditions, but we have very high phosphorus conditions already in the soil. So are they providing some other benefit? And could it be drought protection? The other interesting thing, and I know another researchers have been working on, is can you reduce this mycorrhizal dependence by intercropping, for example, with wheat, with piola? And I don't know, Carol, whether we've had any talks on piola before, but I think that's also an interesting concept. So the pea would be mycorrhizal, the brassica would not, so that you could somehow overcome that. So I think that's all I've got. That'll allow us plenty of time for discussions or questions.